Then next entity, this is the Merkel cell carcinoma. Why I say that low power, we can see this lesion may be in the head and neck, face, or maybe scalp, because we can see the hair follicle deep down in the subq, and then this big bluish nodule. Then when we zoom in, it's really identical to our small blue cell differential diagnosis. The cell is really busy, really hyperchromatic. You can see tons of apoptosis, really easy to find apoptosis. And then also mitotic figure is everywhere, so you know this is really high grade. And that cell with really hyperchromasia and then really hypercellular. Sometimes just almost like molding together, and then didn't form any distinct structure, just this nodule and sheets of cells. But when you have this pattern, you really need to do punch of staining for the diagnosis, because for small round blue cell, we have tons of differential diagnosis. Merkel cell is estimated like the primary tumor of the skin. And then we have other metastases, even like small cell carcinoma from the lung, and then maybe also other neuroendocrine carcinoma from anywhere else. So we need to do the staining. For the classic Merkel cell carcinoma, that is CK20. We have the slide here, that is CK20. The classic pattern is this dot light positive. Of course, you can have cytoplasmics and membranes staining, but the classic one is this dot light, and then CK7 negative. You can also do Merkel cell polyoma virus that is really specific but not really sensitive at all. Around maybe around 60% of Merkel cell carcinoma, you may have that MPV virus positive. So if positive, it is really helpful, but if negative, you could not rule out Merkel cell. And then we can also do TTF1. TTF1 usually negative, but in our lung primary, TTF1 should be positive.